It is my great pleasure today to introduce Dr. Bratislav Misic, who's assistant professor at the Montreal Neurological Institute, a Canada Research Chair, Killian Laureate, an awardee of Brain Canada's Future Leaders in the Canadian Brain Research Program. There's lots of lauds there. Bratislav originally got his PhD with Randy McIntosh and a postdoc with Olaf Sporns. His work really stands at this sort of crossroads of computational and clinical neuroscience, and really the forefront of both. If there's a central goal, there are many goals and many accomplishments already of his career, but it's sort of how, in quantifying how complex behaviors emerge from connections and interactions among brain areas. You know, there's an old quip about the brain, that if the brain were so simple that we could understand it, we would be so simple we couldn't. Bratislav work, I really think it helps sort of solve this paradox. And he does so by engendering these distinct vantage points in almost all of his work. So these vantage points kind of combine, interact, and form. An example of this, there's now a fairly common analytical technique of estimating functional connectivity from structural patterns. And he brings that together with psychoarchitectonic maps, and has shown that, you know, the divergence between structure and function follows some of these psychoarchitectonic, geez, why did I pick that word? Some of these hierarchies. So, you know, in this kind of work, these distinct vantage points still remain in the work. There's no generalization on each of them. And I think each one informs each other in kind of an iterative fashion. So really, quite simply, it allows the complex to remain complex. And it's in this new landscape that I think there's a really a great role for more comprehensive understanding. We might actually be complex enough to understand this complex structure. His work has one of the steepest citation curves I've ever seen. I mean, it's very highly cited, that everyone knows that, but I think it's even more important to say that Bratislav is helping build a field. And not only through empirical work, but also I think in very practical ways. He's has roles in supporting numerous important organizations in the field. He has over 200 reviews registered on Publons, which is insane for me to think about doing that many reviews. And his science is just very open and in an exemplary way. I think the code and data that you see today will be is always available, and I think the methods really are ways of engendering new ways of thinking about how to do science. So I'll stop there. I think we're in for a real treat today. So if everyone could just join me in welcoming Dr. Misic and his talk today on structure-function relations in brain networks.
Thank you so much for that really kind introduction, Simon. Thank you for the opportunity to speak here, and thank you all for attending. I wish I could be there in person, especially since it's snowing right now in Montreal. So believe me that next time I will choose the in-person option. But I'll tell you today a little bit about what we've been doing on this question of structure-function relationships in the brain. It's a question that in one way or another I've been trying to address since I started my PhD. And over the past few years, we've really tried to attack the question from new conceptual angles, and we've been questioning some of the assumptions that we've been making, and specifically what we actually mean by structure and function in the context of the brain. So just to get started, if we take a completely reductionistic approach about how the brain works and how can we characterize interactions between the different components of the brain, we can start all the way at the simplest microscopic scale and think about gene transcription and how genomic gradients are instantiated over the cortical sheet. We can think about how neurotransmitter receptors are positioned and how do they help to modulate signal propagation across a variety of electrochemical synapses at a slightly coarser scale. We can think about different cell types and how they're arranged together. to allow different types of communication to take place and different functions to be implemented. And then for those of us who are in human neuroimaging, we can obviously think about brain structure from the perspective of kind of the large scale white matter and gray matter architecture. For me, for instance, this comes in the form of diffusion weighted MRI. And also we can think about function. Again, for those of us who are in the MRI world, this comes down to bold measurements of the hemodynamic response function, and we conceptualize function in that way. So what I'll talk to you today about, I've kind of broken this up into 3 components. One where we start to ask questions about the relationship between structure and function from the perspective of multiplex communication processes. And there I just mean relaxing some assumptions about what the actual transfer between structural and functional connectivity might be. We'll talk a little bit about annotated networks, this idea that we can actually enrich our representations of the connectome with a variety of biological metadata. And finally, we'll relax the assumption that function can only be measured as kind of functional connectivity in the brain, but can really be thought of more broadly as a set of computations and cognitive processes. And to address this question, I'll show you some of our work using what's called reservoir computing, where we use connectomes to construct and constrain artificial neural networks. So just to begin, to kind of get us all on the same page, we really live in an exciting time because we can now map, image, and trace the connection patterns of many different brains and nervous systems. We can do this in something as simple as C. elegans or in the mouse, but we can also do this in the living human brain. So what used to be done with painstaking manual dissection and post-mortem tissue can now be done relatively automatically using techniques such as diffusion-weighted MRI, where we can reconstruct the white matter fibers that interconnect different neuronal populations and then distill these data sets down into these intricate wiring diagrams that we call connectomes that tell us the probability that 2 neuronal populations are connected with one another and the potential for them to interact with each other. So when we talk about brain networks, we're really talking about 2 different types, and this is a terminology that I'll use throughout. With structural connectivity networks, what we're doing is dividing the brain up into a set of nodes or parcels, and then using some form of either histology or imaging to reconstruct the white matter fibers that interconnect different nodes to one another. This gives us a table or an adjacency matrix that simply has nodes in the rows and nodes in the columns. And any one of these elements tells us the probability that 2 areas are connected with one another. We can do something very similar, but to try to reconstruct functional connectivity. Here we can record from the same brain areas either electromagnetic or hemodynamic activity. And we can correlate time courses that belong to pairs of brain areas. The idea being that if 2 brain areas display coherent fluctuations in neural activity, then perhaps they're engaged in some common function. They might even be exchanging information with one another. So we can go through the same process to reconstruct what are called functional networks, and we can subject both types of networks to graph analysis. The main thing to keep in mind is that with structural connectivity, we're really talking about a relatively stable physical infrastructure that is also by nature sparse. So not all nodes are connected with one another directly. With functional connectivity, we're talking about really a statistical concept. We're talking about patterns of coactivations that are inherently dynamic and can take on multiple configurations across time. And one thing that we know over the past 10 or 15 years is that there is an imperfect correspondence between structural and functional connectivity. First of all, there are many functional connections that are not supported by an underlying structural connection because not all areas are directly connected with one another structurally. And the magnitude of the functional connections that are supported by a structural connection tend to be larger than the magnitudes of functional connections that do not share a direct anatomical connection. This is a fairly common result where if you take pairs of areas and you compare their structural connectivity and their functional connectivity, you see a positive but generally very weak relationship. So some people will look at this and say, well, this is actually really great because I expect them to be correlated with one another. But I think what's more interesting is that there is a lot of variance in functional connectivity that's not directly accounted for by variance in structural connectivity. And the simple reason for this is that areas can be communicating with one another over multisynaptic pathways, so polysynaptically. You could have multiple signals traveling in many different ways over the network to give rise to apparent functional connectivity between two areas. And this is just another example of this concept that even if we were to take what's shown here in blue, these are structural connectivity networks, and then this kind of more, I don't know how I would describe these colors, sort of teal and pink. These are functional connectivity networks. You see that even if you were to try to find modules in the structural connectivity network, they do not actually capture the inherent modular structure of the functional network. And at the same time, if you were to find the modules of the functional network, the so-called intrinsic connectivity networks or resting state networks, they do not very well capture the modular structure of the structural network. In other words, the network architecture of these two modalities is actually fundamentally different. And this is a topic that we reviewed in some detail recently. So what we think is happening is that there is an underlying anatomical infrastructure that we call structural connectivity that supports the propagation of a myriad of elementary signals and signaling processes and communication events. And the superposition of these communication events and signals ultimately manifests as patterns of interregional covariance that we call functional connectivity. So structural connectivity and functional connectivity are two domains that are empirically accessible to us, but the nature of this transfer function of this communication process is something that we have to try to infer. And the way that this has traditionally been done is for researchers to come up with some model of communication and then to test it out on the data that they have. And I'll show you just one slide. This is slightly old work at this point that I did when I was a postdoc, but it's a pretty, I think, prototypical kind of study where we, in this case, we used a very simple percolation model to try to predict functional connectivity from structural connectivity. And in this kind of model, what happens is you have a perturbation. So this represents the transduction of a sensory stimulus, for instance, and that perturbation then spreads via the anatomical network, hitting multiple downstream brain regions. And when you superimpose this kind of process on a structural network, you can simulate lots of interesting, potentially meaningful scenarios, such as how would a single stimulus spread through the network? How would multiple stimuli, so for instance, binaural input, spread through the network? Or what would happen if you had two signals, but they carry different content? How would they actually compete for influence or potentially integrate in the network? So there are a number of inferences that we can make from models like this. We can, for instance, ask, what are the projections that are used most often to propagate these signaling cascades? And you can see here that it's a lot of the connections between the so-called rich club of the brain, a lot of the medial projections. We can ask, what are the areas that tend to be the focal points, the most integrative areas in these kinds of models? And here, what you can see is a number of polysensory association areas that actually correspond very nicely to the default mode network, the dorsal attention network, and the frontoparietal network, that appear to get a lot of their integrated function because of the way that they're embedded in the global anatomy. But what is really important for what I want to show today is that we can use these models to try to predict functional connectivity. So we ran this kind of simulation on structural connectivity, and at the same time, we had functional connectivity in the same participants. So this is the functional connectivity matrix, the empirical one. We can use the model to try to ask what is the probability that on average, the two brain areas co-activate at the same time, because we think that is what functional connectivity represents. So we can use the model to try to predict functional connectivity and then match this to the empirical functional connectivity matrix. And what we've showed here was that on the whole, we can do a decent job. We're about in line with what people have done with much more complex but physically realistic models, such as neural mass models. But it shows you one way of working where we come up with a model that we think operates, for the whole brain, and then we test it against functional connectivity. And we're going purely from structural connectivity just to functional connectivity. And so what I'll show you over the next, hopefully 30, 40 minutes or so, is how we've deconstructed this idea and this way of working and come up hopefully with a new way to approach this question. So I'll start by telling you a little bit about how we can take this into this domain of multiplex communication. And here the starting point is that the study that I just showed you and many others in the field actually make two big assumptions that I don't think are necessarily all that valid. One is that there is a single optimal model that operates on the whole network. It's either contact percolation or it's some kind of centralized communication or navigation or something like this. And the other big assumption is that the transfer function going from structural connectivity to functional connectivity is global in nature. That is that every brain area communicates in exactly the same way to other brain areas. And this really ignores a lot of the regional heterogeneity that we know to exist in the brain. It's something that I'll cover in more detail later. But the idea here is actually, sorry, I skipped this. The reason why I'm showing this figure with cell phones is that in many other types of communication networks, we know that multiplex communication is actually optimal, where you have shared use of a common medium. So for instance, the codomain multiplexing algorithm that is the precursor to what we now have as 5G in our cell phones. But it's also something that's commonly used in broadcasting, and not just in telephony, but in broadcasting, on the internet as well, and so on. So here the idea was that, well, could we potentially take different classes of the communication mechanisms and see whether we can do a better job of predicting functional connectivity, and whether we could do this in a way that was more regionally specific. So that's what we're doing here. These are our kind of two old objects of study, structural connectivity and functional connectivity. And what we did was to say, okay, first of all, I'm going to try to, in order to account for these polysynaptic communication relationships between different brain areas, I'm going to represent functional connectivity as a series of three predictors. And the predictors that we chose here we deliberately chose them because they kind of represent in very broad strokes the three different classes of communication mechanisms that have been proposed in the literature so far. So what we're looking at here is simply the Euclidean distance between brain areas. This would be consistent with a navigation-like mechanism. We're also looking at the path length, so the number of hops on the structural network between pairs of brain areas. And this is consistent with a centralized routing-like communication process. And finally, we're computing something called the communicability, which is a measure that's more consistent with a decentralized diffusion-like communication process. And in order to try to address this question of whether different areas potentially communicate differently, what we're going to do is we're going to take just one region in the network, and we're going to take its entire functional connectivity profile, and we're going to try to predict this functional connectivity profile from its different structural connectivity profiles using these three measures of communication. So what we're really going to do is fit a very simple linear regression model to the functional connectivity profile of every single brain area. And we're going to see how well structural connectivity can predict functional connectivity in different parts of the brain. And probably the most interesting result that we had here was that if you were to look at just the R-squared, the adjusted R-squareds from these different regressions, is here plotting them as a histogram. So the R-squareds for different brain regions that tell us the extent of structure-function coupling, you see that there is a huge amount of variance. In some areas, you have very large structure-function correspondence. And in some areas, it's very difficult to predict function from structure, even though we have three predictors. And when you plot these in a brain, this is what you get. So the way that we plotted this particular figure was to emphasize those areas where you have particularly poor structure-function coupling. So those are the big blue circles here. And what you can see is that these areas are the kinds of areas that we typically think of as being polysensory association areas, or part of heteromodal cortex. So what you have here are the posterior cingulate, precuneus, anterior insula, and so on. And this map we thought looked very similar to this very nice map that was generated by Daniel Margulis some years ago, where he used diffusion embedding on functional connectivity to generate a continuous differentiation of or embedding of areas, depending on whether they were unimodal, primary sensory areas, or more transmodal areas that are shown in red here. And what we find is that when you correlate the position of a brain area in this unimodal transmodal hierarchy with the extent of its structure function coupling, you see this nice negative relationship. In other words, unimodal areas have a very prominent structure function coupling, easy to detect relationship between structure and function. In transmodal cortex, there is very poor structure function coupling. And this was, this shows something very interesting, because what we're seeing is that it is not the case that necessarily all areas act in the same way, and that the same kind of communication model should be applied to all areas when trying to predict their functional connectivity. The other thing that stood out to us here, and this was something that you can see in this beautiful review article from Randy Buckner and Fennekrinen, is that perhaps the way that we're going about this is maybe incomplete, because what they argued for in this TICS paper is that in these transmodal areas, what happens is that due to rapid evolutionary expansion, you suddenly have an untethering from between structure and function. You in these polysensory areas, what you see is a divergence from the kind of strong genomic gradients that you have elsewhere in the brain, and you suddenly see the appearance of much more complex circuit configurations, and one that support more than just kind of simple stimulus response loops and relationships. And so we started to think a little bit more about how we could enrich our representations of the brain. What I'll show you just before I get on that topic is just one more study where we actually tried to understand a little bit better how this gradient of structure-function relationships actually works. So what Vincent Bazinet in the lab did is to run a very simple diffusion process, starting in different parts of the brain, to try to outline for every brain area who its immediate neighbors are over larger and larger circuits. So he could have, for a given area, only its immediate neighbors, or kind of how it's embedded in a slightly bigger circuits, and then how close an area is to other areas in the context of the whole brain. So he's kind of expanding the neighborhood around a particular area in the structural network. And he's looking at what neighborhood size is this area most central? Does this area particularly become important? And what he showed was that essentially the areas that are preferentially central globally when the neighborhood is very large tend to be these polysensory association areas, and areas that tend to be preferentially central locally tend to be a lot of these unimodal sensory areas. But to really focus it on structure-function relationships, what he did was to actually ask at what point, as he tunes the size of these expanding neighborhoods, at what point does he get the biggest match between structural and functional connectivity of a particular area? And what you can see here, so that's what this color bar is showing. It's the diffusion time of this process. So just whether it's a very compact circuit or a very large circuit. And what he shows is that for the transmodal areas, you get the best match at this global scale, and for the unimodal areas, you get the best match between structure and function at this local scale. Again, we can compare this to the Margulis, functional connectivity gradient, and we see a nice relationship between the two, which is to say that there's a lot of regional heterogeneity here where the unimodal areas, as expected, tend to communicate in the context of very circumscribed, very localized circuits, whereas the transmodal areas tend to preferentially communicate over much more distributed circuits. And I'm just going to show you one thing that we've also done to kind of get at this idea of how this hierarchy influences communication. Here we're taking structural connectivity networks, and we're also labeling nodes based on where they lie in this unimodal transmodal hierarchy. So what we can do is actually, for every communication pathway, we can actually say what sequence of nodes is encountered along the way, and we can construct these communication motifs and path motifs that tell us exactly how information travels up and down the hierarchy. I'm not going to go into any results from the study, but I just wanted to point out that we have developed a methodology to kind of look at this in more detail, and I'm giving the reference at the bottom. So those are some of our forays into looking at structure-function relationships, but really focusing more on how these relationships are localized. And what this got us to think about was whether we should be perhaps looking at other features of our nodes that are not necessarily part of our standard connectome representation. And so I'll tell you a little bit about what we've done in this domain. For people like me, this is how we tend to think of the brain. It's a network where we have gray matter nodes that are interconnected by white matter edges. This is what we would get from a combination of standard, structural MRI and diffusion-weighted MRI. But this is an inherently incomplete description, because while this is a very helpful way to represent the brain as a complex system, it's a view that abstracts away a lot of important biological details. So we know that all of these areas are actually different from one another in terms of their gene transcription, site architecture, laminar differentiation, receptor fingerprints, laminar all kinds of things like this, intrinsic dynamics. And a lot of these things we can actually measure in our garden variety MRI scans, and also we have access to from other data sets that are available openly. So what we've started to do is to think about whether we could bring into our models of structure-function coupling some of these more microscale or microstructural attributes. And what I'll show you is just one example where we've done this, not only for structure, but also for function. This is a study that was led by Galia Shafi, where she took the time courses, bold time courses from the Human Connectome Project, and she subjected them to what's called a highly comparative time series analysis. This is a tool called HCTSA that was developed by our collaborator, Ben Fulcher, where you essentially give it a time series and it computes the 6,000 to 7,000 different time series measurements that have ever been invented in the literature. So anything that has to do with the spectral power of the signal, with the repeatability or the autocorrelation in the signal, with variability in the signal, or its entropy, or its actual just amplitude distribution and things like this. And what it allows you to do is to take a particular region in its time course and deconstruct this into a time series phenotype. So for every brain area, we have a kind of a vector of these time series features, and we can do this for every region. And we can do two things with this. We can either correlate the time series profiles with one another to get what we've been calling the temporal profile similarity matrix, or we can take these data and try to see what are the dominant patterns of variation across many of these features. So for instance, applying PCA to this data. This is what one of these temporal profile similarity matrices looks like. So what you have are regions in the rows, regions in the columns, and here we have just how similar the the time series fingerprints or phenotypes of different brain areas are. And in the black circles, you see the standard canonical intrinsic networks. And what you can see is that this network actually has a lot of structure. It does, in some cases, resemble the resting state networks, but there are important deviations as well. What we know is that this temporal profile similarity has this kind of exponential drop-off with distance, which is very similar to what you see in virtually any other type of connectivity data. So areas that are close together tend to be very similar. Areas that are far apart tend to be very different. What we can do is simply remove or regress out this effective distance and then ask, what is the temporal profile similarity for areas that share a direct anatomical connection versus areas that do not? And what you can see here is that areas that are directly connected tend to have more similar temporal profiles. So they are definitely on a much more similar wavelength with each other compared to areas that are not directly connected. And also areas that fall within the same system, within the same cognitive system or intrinsic network tend to have much more similar temporal profiles. So all to say that the architecture of the network leaves an indelible imprint on the actual local dynamics of these brain areas. I'm just going to, and then what we can also ask is how do these temporal features relate to the underlying microarchitecture, which is what we were interested in the first place. So here we're applying PCA to these temporal profiles, and what you can see is the top two components actually have a topography that perhaps is evocative, one that's more of a ventromedial dorsolateral kind of gradient, if you will, and one that looks very much like this unimodal transmodal hierarchy. What we can ask first is what are the actual dynamics that contribute to this differentiation of brain regions? And what we see is that for the first component, it really captures regional variation in the repeatability of the signal. So what we're plotting here is just the simple lag-1 autocorrelation as well as the nonlinear autocorrelation of these time series. You can see that the maps look very similar to this overall component. These are the kind of the most highly loading features. You can see as an example how different time courses from areas that are at different parts of this gradient would look like. And then for this second component, what we find is that the time series features that actually load most highly are those that have to do with how evenly distributed the signal amplitudes are around zero. So what we saw was a lot of measures of distributional entropy and a lot of things that a lot of the even moments of this signal amplitude distributions. So things like kurtosis and so on. What you can see here, these are the actual signal amplitudes that you encounter, and areas that are shown in red. So the unimodal cortex, for instance, tend to have wider tails here, which means that they can actually take on a bigger range of values compared to their transmodal counterparts. In other words, in unimodal areas and sensory areas, you have a wider range of values, presumably because you have to map different types of stimulus variations in those areas. But what was interesting here was then, well, if we see that the actual local dynamics are different, can we map this onto different types of circuit properties? And what we did in this particular study was to relate these gradients to the first component of gene expression from the Allen Human Brain Atlas, to this unimodal transmodal hierarchy, to this T1 T2 ratio, which is a proxy for intracortical myelin, and cortical thickness all in the same participants. And what you see is that this gradient of kind of intrinsic time scale and repeatability is actually highly correlated with the first component of gene expression, whereas the second kind of unimodal transmodal variation in the distribution of signal amplitudes looks very much like this unimodal transmodal hierarchy, as well as the T1 T2 ratio. In other words, because the underlying microarchitecture of these areas is different and the circuit configurations are different, you start to also see different types of dynamics emerging. And perhaps we shouldn't always just be looking at functional connectivity over the course of a whole time scan, but we should be going back to the basics and asking whether we can actually see the effect of structure just on the temporal dynamics first. What you've seen me do just now, if you were paying attention to some of these p-values, this is just an aside, but you would have noticed that we tend to do what's called a p-spin here. So these are so-called spin tests, and this is something that the comparison, kind of correlating pairs of brain maps, is something that's become very pervasive in our work, but also elsewhere in the field. The problem is that brain maps derived from MRI or virtually any other imaging techniques are highly spatially autocorrelated. So areas that are close together tend to be very similar to one another. This creates a problem when you're correlating two brain maps because the points in that scatter plot are not independent from one another. So you tend to get, if you use kind of standard statistics, you tend to get very inflated p-values. So there's been a range of null models that have been proposed, notably those by Aaron Alexander Block and also Josh Burt, that tried to account for the spatial autocorrelation. So these are so-called spin tests. And at the time that we started doing this, there were at least 10 different ways of implementing them in the field. So I just want to, if you're interested more in this particular topic, Ross Markello actually took a very deep dive into this. He implemented all of these different spin tests. He's got the code and the walkthroughs here. And he benchmarked all of them in terms of how well they can control the false positive rates and how well they behave in different types of situations. So I'm not going to go into any more detail than that, but just to say that we do kind of show which ones tend to be best and which tend to be more appropriate for different types of experimental questions. So this, I showed you just an example of how we can look at microstructure and how it influences function. I just want to take one slide to show you with probably the most extreme example of this, where we said, well, how can we bridge all of these different scales of descriptions? And what we did here was to say, well, in our mind, at least what we can measure, the most microscopic scale is gene transcription, and the most macroscopic scale is function as defined, by cognitive function. And it turns out that we actually have two really wonderful atlases of both that are freely available. And what Justine Hansen in the lab recently did was to take maps of gene expression across the brain from the Allen Human Brain Atlas. So this is microarray expression that was made available by the Allen Institute, and to try to map this to patterns of functional activation that are available in Neurosynth. So a wonderful tool made by Collier Coney that tells you the probability that a particular area is activated in the context of some cognitive term. So what she did was to get these two data sets and to map them to one another using a technique called partial least squares. And what she found was that there was this one single variation across the brain, really a genetic signal that could robustly separate cognitive processes from affective processes. So that's what I'm going to show you here. This is what you get on the kind of the cognitive side. So you have areas that tend to be more associated with affect. So you have things like fear, emotion, stress, and things like this, valence, and areas that tend to be more associated with visuospatial attention and so on. So visual attention, imagery, skill fixation, things like this. And concomitant with that is a genetic signal that actually separates different types of genes from one another. These are the kind of pathways that you might get from a standard Go analysis, but what I think is really interesting is that you can get the resulting gene signal and you can do cell type deconvolution. So you can ask what types of cells tend to be enriched in areas that are more specialized for affect versus areas that are more specialized for these kind of more cognitive or attention-based processes. And what you find is that essentially, to cut a long story short, it is the inhibitory and excitatory neurons that are enriched in these areas, in areas that have to do with cognition, and it is more the support cells like astrocytes and microglia that tend to be more enriched in areas that have to do with affect. In other words, what we have is a mapping that really goes from all the way from the most microscopic scale to the level of kind of psychological processes. And I think it's a nice example of how you can potentially bridge scales, and but obviously there are a lot of intermediate levels that we have to fill in. We do explore this in some detail, exactly how these mappings depend on site architecture and things like that. And you can take a look at that in Justine's work. We also show that this relationship strengthens in development, and we can replicate it in lots of different data sets. Just another side note, what you've seen me talk about twice now, at least, is this idea of measuring gene expression in the brain. We have actually developed a tool that you could readily apply in your own work. It's called ABAGEN, Allen Brain Atlas Genetics. So it was developed by Ross Markello as well, who it's basically a very lightweight Python toolbox that at its core is really just one line of code. In that line of code, you give it the atlas, your parcellation that you want. It'll fetch the data from the Allen server. It will do a number of pre-processing steps for you. It has defaults that we recommend, but you can also implement lots of different things. And it'll spit out this gene expression table that you can then use to do lots of different analyses. And it takes really just 1 minute to run approximately. It gives you lots and lots of different options where you can either get specific genes of interest, you can parcellate in lots of different ways. It works with volume of surface-based atlases. It can interpolate maps and so on. The most fun part of this is that it will also write your method section for you, depending on the methodological choices and the pre-processing steps that you choose. And this is our way or effort to try to get the field to maybe adopt more standardized reporting, at least in the context of imaging transcriptomics. So if you're interested, you can see Ross's paper or go to the website that we've created. So that's us going from gene transcription all the way to function. Out of all the things that you see here on this kind of cartoon layout, I would say that arguably the one that the modality that I'd be most interested in using in my own work for looking at the relationship between structure and function is the distribution of neurotransmitter receptors. Neurotransmitter receptors are very important, as we all know. Both the cortical and ascending projections, obviously they modulate both of those types of interactions, and they can, theoretically they can modulate the signal such that you have profoundly different functional network configurations. So you can either have projections that promote more segregated processing or more integrated processing. The actual receptor distribution is something that we actually can measure with PET. There are lots of different tracers that can tell us about the brain-wide distribution of different neurotransmitter receptors, but there is no atlas of what these of how these receptors are actually distributed. So sometime last year around Christmas, Justine Hansen in the lab and we started to cold call anybody that we knew that had PET data and was willing to share it. And this took lots of meetings, but the end result, which we're showing here, is a collection of lots and lots of different PET maps that have all been kind of processed appropriately to that particular radioligand that are the control groups of various studies. So these are all in healthy participants. And these are the maps that we have so far. There's actually one missing because we've recently added NMDA, but long story short, what we have is about upwards of 1,200 PET maps, so 1,200 participants that cover 19 different receptors and transporters and across 10 different neurotransmitter systems. You can get the maps and the code here on our GitHub. And we've done kind of a few different things with it, but I just want to show you something that's very relevant to this question of structure function coupling, where we can actually take these receptor maps And for every pair of areas, we can ask how similar the receptor profiles are. And we can see whether this information from the neurotransmitter receptors and transporters can actually help us to make better prediction, how well it aligns, first of all, with structural and functional connectivity, but then also if it can help us to predict functional connectivity from structure. So what you're seeing here is that areas that are directly structurally connected tend to have much greater receptor similarity. So they are much more likely to kind of engage in common communication channels and to be co-modulated with one another. We see that areas that fall in the same functional network also tend to have much more similar receptor profiles. So different cognitive systems tend to have their own unique receptor profile or receptor fingerprint. And what we're doing here is we're trying to predict function from structure in exactly the same way that I showed a few slides ago, using different predictors. But here we're just doing the prediction of function just from structural connectivity. And here we're predicting function from structural connectivity and from receptor similarity. And what you can see is that by adding information about the receptor similarity of different areas, you actually get a boost in prediction. So, and these are the areas that actually benefit most from adding this information. Is this because in these areas we simply have a poor measurement of structural functional connectivity, or is this telling us something about how these areas communicate above and beyond just the information that's in structural connectivity? I mean, we can't quite tell that, but what we can say, I think quite confidently, is that incorporating more of these biologically meaningful features actually helps us to predict function from structure. So those are some of our recent attempts to come up with richer representations of structural and functional connectivity. And just in the last maybe 5 minutes, if I can, I'll show you how we've gone about rethinking not just structural connectivity, but also functional connectivity, and what we actually mean by function. In connectomics, if somebody says function to me, I automatically think of functional co-activation, so correlating time courses and things like this. And like I said, a lot of the field has really focused on just predicting functional connectivity from structure. But what we really want to understand is how the underlying network structure actually supports computation and cognition. So what Stephanie Suarez in the lab did was to take a technique from artificial intelligence called reservoir computing and to apply it to connectomes. And what happens in reservoir computing is you have an artificial recurrent neural network that tries to extract information from a continuous stream of external stimuli. So you have an input layer and an output layer, and you simply put in some input signal. The signal propagates through the network. It gives rise to complex patterns of neural activity in this particular reservoir, and the activity time series of these readout nodes is then used to train a linear readout unit that reproduces, in our case, a delayed version of the signal. So what it is just a paradigm to take any arbitrary connectivity structure and teach it to do different kinds of tasks. And if you look at that literature, mostly people will use completely random reservoirs, and you can also have your own kind of choice of arbitrary dynamics to superimpose on top of this. So what Stephanie did was to take connectomes, implement them as reservoirs, and then teach them to do this particular task. In this particular task, what we're doing is training the reservoir to learn to reproduce a delayed version of the input signal. So it's really just a you can think of it as a kind of a memory task. The signal comes in, the reservoir, the dynamics of these nodes represent the signal in some particular way. Then we read out that signal from the purple units, and we train a classifier to reproduce the delayed version of the signal. So you can really think of this as a working memory task, And what we can do is also tune the dynamics of the reservoir such that we go from very simple kind of stereotyped dynamics all the way to critical dynamics and then chaotic dynamics. So in this regime here, as you can see here, we have kind of stable dynamics. Here we have chaotic dynamics, and somewhere here we have at the edge of chaos or at criticality. Well, that's what how we would describe this particular regime. And the parameter that we're using, we can just tune this with these dynamics with with just a single parameter. So it allows us to control both the network structure, but also the dynamics, and ask how well the network does. So the first thing that we wanted to ask was, well, I'm just going to say that if you take a look at the paper, you'll see that we actually do consider in some detail exactly how different cognitive systems can actually reproduce these signals. But what I just want to show you is kind of the key result, and the last thing I'll show is that for a range of these criticality parameters, we can look at the memory capacity of our network, which is shown here in purple, as well as a population of rewired null networks. So we take pairs of edges and we randomly flip them. And what you can see is that when the dynamics are critical, which has often been in different literatures, has always been posited as kind of the working point of the brain, you see that the connectome actually does better than this population of rewired null. So there's something about how the network is, how brains are wired, that supports optimal, in this case, working memory capacity at this critical point. What we then looked at was to say, well, the rewired networks, so they do more poorly here, but sometimes they actually do better in the other regimes. So the question is, well, is this really a fair comparison? Because what happens with rewired networks is that you can randomly move edges around, and you can place edges between areas that are much further away. So for instance, the connection length distribution of the real brain is shown here, and the connection length distribution of these null models is shown here. You can see that they tend to have much longer connections. So they have this unfair advantage. So if we were to appropriately normalize our measures of memory capacity by accounting for this extra wiring cost that rewired nulls have, you actually see that real brain networks outperform rewired nulls across all parameter settings. So this is interesting because, for us in neuroscience, it's interesting because it says that there are some properties of the connectome that seem to be important for this optimal performance. We show in the paper that it has a lot to do with the modularity of the network. The other way that this is important is that it means that you can actually build better AI, and you can actually build more efficient physical hardware and chips, what's called neuromorphic hardware, by implementing some of the organizational principles of real brains. So that's kind of all I wanted to show. I hope I've convinced you that network structure seems to shape dynamics and promotes a whole variety of behaviorally relevant communication patterns, and that there is a lot of regional heterogeneity that we've previously been blind to, but that supports kind of this interplay between local attributes and global dynamics. And that we and others in the field should perhaps take a step back and really rethink what we mean when we say structure and function in the context of this problem, and that I think there's a lot of benefit to considering multiplex communication processes with richer dynamics, and also to focus on annotated brain networks that perhaps involve a lot more meaningful biological metadata, and also to perhaps consider not just the functional connectivity that we get in the brain, but also the computational capacity of the brain. So I just want to thank you for your attention, and thank a variety of people who've contributed to this work. I just added the slide on the transmitter receptor, so I haven't added everyone from that. I apologize for that, but I'd be happy to take any questions.
Wonderful talk. We try to lead with questions from trainees. Is there anyone at DIBS or online that would like to ask a question first? We have just a couple minutes. So, amazing talk. A question I just sort of had is something that's really cool about the reservoir computing is it estimates an activity pattern. And one of the kind of barriers between the world of task-related cognitive neuroscience and a lot of some of these computational approaches is that is the HRF, right? Or the estimation of an event isn't really taken into account in a lot of ways. But something this kind of seems to do is maybe even be able to estimate what an event-related pattern or if I guess my question is, do we get to the level of events when we are trying to estimate this behavior, or could this be a sort of sub goal of this kind of?
Yeah, so the reservoir at its core is a dynamical system. What we did was to use very simple kind of threshold units, but you could use something more complex. Actually, the way that we first started to do this was to use neural mass models from the virtual brain, so you can make the underlying dynamics very biologically realistic. But the point is you have a lot of freedom, and all of these units have time series that you can save and recover afterwards if you want. And then you could, if the underlying patterns are, you can either pull them through kind of a forward model if you wanted, like a balloon model to generate hemodynamic activity, or you could simply correlate them as is and generate functional connectivity as well. We did it a little bit, but I think there's actually a very nice space for people to explore there still.
Yeah. Andrew, you have a question?
Yes, Simon, I'm happy to wait because I tend to ask questions all the time. If somebody else, if not, it's great talk. Thank you so much. I have a lot of technical questions, but I'll spare those. But more on a kind of philosophical level, it's really interesting how you showed that the gene expression was highly, kind of not highly, but somewhat significantly correlated to functional connectivity. And I believe you measured connectivity based on resting state data. Is that?
Yeah. Yeah.
Okay. So so my question is, there are various forms of functional connectivity. You can have resting state connectivity, but then the connectivity can change a little bit when somebody does a function. There are dynamic properties of the functional connectivity. Then there is also various methods to compute functional. So in spite of all these variability, you're able to show, you're showing that there is a strong correlation, for example, between gene expression and functional connectivity. What does that mean from a very high level? It's like, does it mean that the resting state can very well capture all these variabilities, or are we missing something?
It's a great question. I mean, so one thing is that particular result was not functional connectivity specifically. Yeah. It was resting state activity, and then what we did was to compute measures of in that case, it was autocorrelation of the signal. So it's something that folks have described previously as the intrinsic timescale, for instance, of the brain. This idea that some parts of the brain are more attuned to very rapid changes in external stimuli, and others tend to be more responsive to changes that take place over longer, more protracted timescales. And then the question is, how does that relate to gene expression? That principal component of gene expression is, taking 20,000 genes and then taking kind of the dominant pattern of variation. That particular spatial pattern appears time and time again. Not just for gene expression and for these dynamics. You can also see it in the neuroscience atlas for these kind of functional associations. You can see it in various cytoarchitectonic data sets. We see it in patterns of receptor profiles. It probably represents a genetic signal that encodes for different cell types. That's what I think that's like the least controversial answer. And this variation in cell types is then going to influence exactly how signals are exchanged locally, how areas can synchronize with other populations, how they're modulated by subcortical projections and so on. So that's why I think these intrinsic dynamics vary in relation to gene expression, but then also why lots of other things that we seem to be measuring about the brain also vary with that pattern of gene expression.
Thank you.
Thank you so much. If everybody could just give another round of either virtual or actual applause. Just fantastic talk. We'll stop there to give enough time for our trainees to now meet with Dr. Misic. And just thank you again.
Thanks a lot.